All right, so we discussed the difference between simple and facilitated diffusion. What's the difference? Yeah. Okay. Okay. Osmosis is when water moves from an area. So if you look at this here the pores are too small for who to pass? Okay. If you look here, guys, these pores, side A and B, the pores are too tiny to allow who to pass through them? The solute, who happens to be glucose. So what does water end up doing? It goes to the site where there's solute. So you see the two levels are going to be like that. You see water move to dilute that part. Now you need to understand the difference between hypotonic, hypertonic, and isotonic solution. Now, the best way to understand hypotonic, hypertonic, and isotonic solution is when let's examine that we have three beakers. We have beaker number 1, beaker number 2, and beaker number 3. And you put fluid solvent in all of them. So the solvent is the major part. And then you put in the middle a red blood cell. You put in the middle a red blood cell. So that's my red blood cell. What's the osmolarity of the red blood cell? 300 milliosmoles. Red blood cell is 300 milliosmoles. 300 milliosmoles. In solution A, you dip the 300 milliosmol red blood cell in a 300 milliosmol solution. So now they're equal. So now what happens to the water movement? Is water movement going to move inside the balloon more, or is it going to move out more, or is the relationship going to be equal? Equal. So here that's an example of an isotonic solution, because the osmolarity inside and outside are equal. Now if the red blood cell is 300 and the solution is 30 milliosmoles, now they're not equal. So what's going to happen? Is water going to go from inside the red blood cell to the outside, or is water going to go from outside to the inside of the red blood cell? Yeah, from inside to the outside. What did we say water do? The water moves from where? From an area of a low solute to an area of high solute to dilute it. Remember I told you, if water in the middle, on this side 300 millimoles, on that side 700, water is going to move to the 700. So water here is going to move more to the inside. So what happens to this red blood cell when so much water goes inside it? Like a balloon. It's going to burst. It's going to burst, and that is known as turgor. That is known as turgor. That is known as turgor. Now, if you look at this solution here, if you look at this particular third solution now, if you examine the third solution, 300 on the inside and 3,000 on the outside. So what's going to happen? Is water going to go from inside to outside or from outside to inside? So water is going to go from inside to outside. So here it bursted because the red blood cells swell. But here water went out. So what happened? Shrink. It's shrinkage. So that's an example of isotonic. This is an example of a hypotonic solution. And lastly, we have a hypertonic solution. This is very important for you guys to know. Is that clear? Okay. So let's now. No, that's on recorded. You're going to see it online. Okay? It's okay. It's okay. It's okay. Don't worry. Where did you get that one from? Oh, I see. No problem. Okay, guys. So let's now back to the PowerPoints. Okay. Okay. Let's look here. So we have a so here you see a hypotonic solution. Water moves in. Isotonic, it doesn't change. Hypertonic, it shrink. Now, the transport maximum is a concept where all of the solute particles have been transported through the plasma membrane, and now we have reached equilibrium. We have reached now equilibrium. That's facilitated diffusion. You see what happens in facilitated diffusion? As you see here, in facilitated diffusion, we see that a lot of particles are on one side, a little on the other. So the diffusion pressure is from out to in. But those particles are so big, number one, to pass through the plasma membrane. Number two, they are polar. They cannot go through the non-polar leaflet. So they need a facilitator. They need a carrier protein that will bring them down. So the solute will bind to it, and the carrier protein is going to send it out. Now, sometimes we have a secondary active transport. What's the secondary active transport? A secondary active transport is when we rely on a gradient that is already there in order to shuttle the movement of another particle. So for example, if somebody is driving to come to school and you take a ride with them, that's secondary transport because you took a ride with somebody. You see these two arrows here. The first arrow is representing the sodium particle that is moving down gradient anyways. And with it, who's taking a ride? Glucose. That's secondary active transport. And then down here, when we use ATP in the sodium potassium ATPase, that's not secondary, that is primary active transport. You need to know the sodium potassium ATPase. The sodium potassium ATPase, okay, it's very important for us to know the sodium potassium ATPase pump. This is an enzyme. Number one, it works uphill against the gradient. If it wasn't working against the gradient, we would not need ATP because when you flow down concentration gradient, you don't need chemical energy in the form of ATP. You know, diffusion is when the particles move from an area of a higher concentration to an area of a lower concentration, powered by kinetic energy, energy of bombardment of the particles, energy of heat. But when you look here, orange represents sodium. Blue represents potassium. Is there more sodium on the outside or there's more sodium on the inside? There's more sodium on the outside. So naturally the diffusion of sodium is to come from outside to the inside. So what does the sodium potassium ATPase do? It will take it and throw it right back out where it came from. Imagine there's all of us now. They open the door. Are we all going to move in or are we going to all go out? So that's simple diffusion. Actually, that's facilitated diffusion because we didn't go through the wall. We went through the door. That's facilitated. Now imagine there's somebody gigantic out there, like, the rock, the rock. So now we walk out. If three of us go out, the rock will take us and throw us right back in where everybody is. Can you imagine that? You know the Black Friday when people are trying to run in? Imagine there was somebody in there blocking them, and if three leak in, try to throw them out. That's the sodium potassium ATPase. And it does that to two electrolytes. When sodium move in, it'll take three and throw them out. And look at potassium. Where is potassium supposed to? Is a lot of potassium inside or outside? So when potassium flows outside, it'll bring it back against the gradient. So is that active or passive? That's active. So you must remember that. Sodium, potassium, ATPase, and obviously it needs ATP energy to do that. It needs ATP energy to do that. If it doesn't have ATP, it cannot do this very powerful job. So you need to know two things. Sodium is more on the outside. When it leaks in, the sodium, potassium, ATPase will throw three of them outside. Potassium, if there's potassium a lot on the inside and it flows out, it'll bring it right back in, but two of them. You need to know this. Okay? This phagocytosis, when we gallop and swallow, you see this is the white blood cell. It's galloping and swallowing a particle with this. Remember the pseudopods we discussed, the extensions, the appendages of the cell at the cell membrane in the interface, and then bringing in a lysosome. A lysosome is full of enzymes, lysosomal enzymes, hydrolytic enzymes, degradative enzymes, metalloproteinases, reactive oxidative species, acid to degrade all of these particles. How do we bring stuff inside the cell? We bring it through endocytosis by forming a clathrin-coated pit. And the clathrin-coated pit, you see we made a pit with all the particles that we need to bring inwardly. Transcytosis is when you leak right through the walls of the cell. That's transcytosis. Exocytosis is the opposite. Is when you throw particles outside. When you push particles outside, and this, endocytosis, endo, bringing inwardly. Exocytosis, exo, throwing outwardly. Is that clear? Now we finish with the plasma membrane. This is the cytosol and the cytoskeleton. The cytoskeleton, you must know that this is the structural framework of the cell. The cytoskeleton, this is the most powerful part of the cell that holds the cell integrity, that maintains the integrity of the cell and give it tensile strength. So the cytoskeleton is made up of proteins. Remember earlier we talked the cilia has proteins like actin and these proteins, the microtubules. So those are also used to give the cell its structural strength. Microtubules have these particular dynein arms, and the nucleus now is the last universe we're going to discuss, because the nucleus now, it has an outer envelope. You see, that's the nuclear envelope, and in the middle it has a nucleoplasm. It's like the cytoplasm, but we call it a nucleoplasm. It is full and rich with the chromatin, the DNA. Inside the nucleus, you have the nucleolus. It has the nucleolus. So that shows you that the nucleus has a lot of holes. So a lot of holes meaning a lot of movement going to be taking place. Things coming inside the cell, things coming inside the nucleus, things getting outside the nucleus. So if you look here, that's the structure of the nucleus. It's attached to the nucleus, and you have a nucleolus. This is where the ribosomal RNA is made. You see these holes? This is how the mRNA transcript is going to leak out. And sometimes you're going to have receptors that bind the cholesterol-based hormones, testosterone. Remember we said it enters the cell, then it goes through the nuclear pore. This is the endoplasmic reticulum, and the endoplasmic reticulum is is it has a smooth endoplasmic reticulum, and there's a rough endoplasmic reticulum. Why does the rough endoplasmic reticulum called the rough endoplasmic reticulum? Because it has ribosomes attached to it. Excellently. What about the ribosomes who are attached to the rough endoplasmic reticulum? Compared to the free, we have two types of ribosomes. Some ribosomes attached to the rough endoplasmic reticulum, and some ribosomes are free floating, swimming in the cytoplasm. So what's the difference? Why do we have two kinds? Why not just one? Why are there ribosomes on the rough endoplasmic reticulum? Why not ribosomes on the Golgi body? Why not ribosomes on the nucleus? Why not ribosomes on the lysosome? Why no ribosomes on the plasma membrane? Why it's gotta be just the rough endoplasmic reticulum? There's a reason. The rough endoplasmic reticulum is gonna, ribosomes in the end, they're gonna be making proteins. Ribosomes in the end, they're gonna be making proteins. Whether they are attached to the rough endoplasmic reticulum or whether they are free floating. In the end of the day, whether you are on the rough endoplasmic reticulum, whether you are free floating, in the end of the day, you're gonna be making proteins. But the proteins that are made on the ribosomes, on the rough endoplasmic reticulum, the rough endoplasmic reticulum is going to stick a sticker on them, a sequence, signal peptide sequence. When the Golgi body reads that sequence, it knows this protein is to be shipped out internationally. It must go outside the cell. It must leave the cell. But the smooth endoplasmic reticulum doesn't make any proteins. The free-floating ribosomes, the protein they make is just going to be used locally inside the cell. See, that's the rough endoplasmic reticulum. The smooth endoplasmic reticulum, important for lipid detoxification. Those are cisterns. This is the Golgi body. Looks like a pita bread. And the rough endoplasmic reticulum sends out proteins that go through the cisterns, that go through the cis side and emerge and exit through the trans side. So the Golgi body has two sides. Cis side to bring in the material, and trans side is to exit the material. Peroxisomes from peroxide, they are neutralizers, they detoxify. So something that is really, really dangerous and toxic like peroxide, why do we put peroxide on the wound? To kill the bacteria. So imagine you have peroxide in your body, it's going to kill your own cells. So you have this peroxisome that will take peroxide and convert it into water and oxygen. So recycle it from something bad and dangerous like peroxide into something that's really nice, known as water and oxygen. Water and oxygen for aerobic respiration. This is my favorite. That's the proteasome. What's a proteasome? Proteasome is the shredder of all the proteins that are improperly folded, misfolded proteins, damaged proteins, defected proteins. All of these proteins are gonna make stress. They're gonna make metabolic stress. They're gonna make a defect on the brain. You know the disease, mad cow disease? Mad cow disease is when misfolded proteins are deposited in the brain. Now, people who smoke cocaine, people who snort heroin, there's a big problem because the rate of the misfolded proteins in their brain is going to increase severely. And your body is already prepared for that kind of misfolding. So your body has systems that will repair, that will shred these proteasomes. You see, every protein that is useless, not useful, harmful protein, there will be a ubiquitin tag. You see this purple ubiquitin tag? If there is a ubiquitin tag on the surface of this bad protein, the proteasome will destroy it. So all of us right now, we have misfolded proteins in the brain, but no problem because the proteasome will shred them. But if you go around snorting heroin and cocaine, the rate of these misfolded proteins is going to go up or down? Is going to go up. And the number of proteasomes and the systems that are rescuing you is going to increase or decrease. So there's going to be a large turnover. So it's like mad cow disease. That disease is called Creutzfeldt disease. But this is just to bring your attention to the proteasome. The proteasome is going to shred and degrade all the bad protein after and only after they have been what? Tagged by this ubiquitin tag. You don't need to know it's ubiquitin, but it is ubiquitin tag. So if you're a bad misfolded, you will have a tag on you. That means you have to be destroyed. Questions? Any questions? The mitochondria is the powerhouse of the cell. We know that from high school. That's the powerhouse of the cell. Through oxidative phosphorylation, aerobic respiration, oxygen, oxygen. If there's no oxygen, that's why hypoxia kills the cells. Why do people have stroke and heart attack? Because no oxygen to the tissue, no oxygen to mitochondria, no ATP, sodium, potassium, ATPase is not going to get paid. They're not going to get the ATP, so it's not going to do its job. All the other enzymes that rely on ATP, they're not going to get ATP. They will not do their jobs. All the activity is gonna shut down, and the cell is gonna die. That's how important the mitochondria is. This is the mitochondria. The mitochondria, they say there is an endosymbiotic theory where you had a prokaryotic cell that swallowed and galloped another prokaryotic cell, but the prokaryotic cell inside didn't die. It was able to evolve a function that is helpful to the bigger cell that swallowed it. That's why you have the mitochondria with two cell membranes. One, an outside extracellular mitochondrial membrane and an inner mitochondrial membrane. And the mitochondria has its own circular plasmid DNA. Yes? Yeah. The proteasome. Mm-hmm. So when the proteasome takes them as misfolded damaged proteins, it will shred them so tiny into really useless, harmless subjects. So your body now, your cells, they have a mechanism of packaging and removing and properly disposing all of these toxic substance outside of the cell through exocytosis. Remember we talked about endocytosis bringing. Exocytosis throw out, and they will throw out in the bloodstream. That's why your blood has red blood cells, it has white blood cells, and it has also plasma. Plasma is full of water, is full of nutrients, is full of also waste. And we want it to be full of waste because blood is not only bringing good things, blood is flushing from the tissue all the toxic waste. Are we clear? It's going to go to the blood. And the blood is going to depend on the waste now. We have different types of waste. Some waste is going to go to the urine. Some waste is going to go to the lung to be expelled out. Some waste is going to go to the digestive tract to be secreted. Okay? Some waste is going to go to the skin to dissipate through the skin. So that's the structure of the cytoskeletal protein known as centrioles, and we will see in cell division they are important in pushing the cell far apart to the two opposite poles in order to allow for the cleavage furrow to happen and in order for the two cells to now divide. So we need those cytoskeletal mechanical proteins to help in cell division. Any questions? Does anybody have any question? Yes,